The 647th Assembly of New Asia College of CUHK 2021 to 2022 will start now. Today's assembly is the second lecture of the 6th Yang Guoyong Lecture in Life Sciences, jointly organized by New Asia College and the School of Life Sciences. We are pleased to have Professor Dennis Lowe Yuk Ming sharing with us translating scientific discoveries into clinical impact. 由科學發現至臨床影響. We are also pleased to have Professor Chen Sun On, our college head, to introduce the guest speaker and to be our moderator at the Q&A session right after the lecture. Professor Chen, please.
Professor Lowe, honorable guests, fellow alumni, colleagues and students, good evening. Welcome to the 6th Renguorong Lecture in Life Sciences. This lecture series is co-organized by New Asia College and School of Life Sciences since 2015, with the generous donation from the family of Dr. Ren Guorong, founding chairman of Department of Biology of New Asia College. Each year, we invite a distinguished scholar to visit our college and to share their experience and accomplishments in pursuit of excellence in life sciences research with our staff, students, and general public. Today, we come to the second lecture. It is my great honor and pleasure to introduce Professor Dennis Lowe. Professor Lowe is currently the Associate Dean in Research of Faculty of Medicine, Chairman of Department of Chemical Pathology, and Li Ka-shing Professor of Medicine of Chinese University of Hong Kong. Professor Lowe started his preclinical studies at the University of Cambridge and completed the clinical training at the University of Oxford. He then pursued training in medical research and obtained the Doctor of Philosophy and Doctor of Medicine degrees from the University of Oxford. After graduation, he was appointed as University Lecturer in Clinical Biochemistry and Honorary Consultant Chemical Pathologist at John Radcliffe Hospitals, the teaching hospital of the University of Oxford Medical School. Professor Lowe returned to Hong Kong in 1997. In the same year, he discovers the presence of cell-free fetal DNA in maternal plasma and has since been pioneering non-invasive prenatal diagnosis using this technology. In the area of cancer detection, Professor Lowe has pioneered a number of approaches to cancer liquid biopsy, especially for the detection of nasopharyngeal carcinoma, and genome-wide approaches for screening multiple types of cancer. In recognition of his research, Professor Lowe has been elected as Fellow of Royal Society of London, Foreign Associate of US National Academy of Sciences, Fellow of World Academy of Sciences, and founding member of Academy of Medicine of Hong Kong. Professor Lowe has won numerous awards for recognition of his achievements and contributions to medical sciences, which include the King Pfizer International Prize in Medicine in 2014, the Future Science Prize in Life Science in 2016, and Fudan Zhongji Science Award in 2019, and very recently, the Breakthrough Prize in Life Sciences and the Royal Medal of Royal Society of London in 2021. May I now invite Professor Dennis Lowe to deliver the second talk of Ren Guorong Lecture on the topic of translating scientific discoveries into clinical impact. Professor Lowe, please.
Thank you very much, Professor Chen. I'm most grateful to New Asia College and the School of Life Science for giving me this opportunity I'd like to share with the audience some of the experience that I have with regard to translating scientific discoveries into clinical impact. So my primary area of research is prenatal diagnosis, which is now an established part of modern obstetrics care. So when a woman is pregnant, she of course is very anxious to know about the health of the baby. But if we want to test, for example, the fetal chromosomes or the cells, then we have to obtain some materials from the baby for testing. And to do this, we have to insert, for example, a rather long needle into the uterus in a process called amniocentesis. But then every time we do this, we have a risk of actually harming the baby or even killing the baby. And because of this reason, for the last 50 years, many scientists around the world has been dreaming about the possibility of doing non-invasive testing. Now, for example, is it possible by testing a blood sample from mother that we could test something about a baby? Now, but this thinking is actually against conventional wisdom because we all know that our blood circulation system and our mother's blood circulation system are separate. So I've been thinking about this area actually since I was a medical student. And then in 1997, I was thinking, is it possible the baby might actually somehow release its DNA into the bloodstream of the mother? But the question is, how do you prove this? So I was thinking of a situation where the baby is a boy. If my thinking is correct, then the baby boy will start to release the male chromosomes into the mother's blood, the Y chromosome. So I did an experiment whereby I take some plasma or serum from the mother, and then we heat the sample, just boiling for 5 minutes, and take a few drops of that boiled soup to do PCR, the polymerase chain reaction, which is a method to amplify DNA, just like we're now using PCR to detect COVID. So interestingly, I actually find that some women have a signal of the Y chromosome, and then some women don't have a signal. And later on, I was delighted and also excited to see that all the women have the Y chromosome, all have baby boys, and all those without signal all have baby girls. So this is the first demonstration that maternal plasma and serum have fetal DNA. And it hasn't been discovered before because previously the dogma is that the DNA should be present inside cells. So you would not expect the plasma to have fetal DNA. And then actually we find that this fetal DNA enters the maternal circulation very early on, just several weeks of pregnancy, and we can see it. And actually by the 10 weeks of pregnancy, the fetal DNA concentration reaches 15%, which is amazing because imagine the baby is so small compared with mother so big. And yet the baby is working so hard to push all this DNA in the mother's blood. So then I'm wondering, is it possible that I can actually use this fetal DNA to do some molecular testing? Now, the number one reason why pregnant women go for prenatal testing is because they are worried that the baby might have Down syndrome. So what Down syndrome? So normally, if you look inside the DNA of our cells, you find that DNA is distributed on chromosomes. And we have 23 pairs of chromosomes. And the chromosomes are numbered so that chromosome 1 is the longest, and 2 is a bit smaller, etc. And then for every pair of chromosomes, we have 1 copy from the father and 1 copy from the mother. And then if you have a boy, then we have XY. If you have a girl, then we have XX, the sex chromosomes. Now, interestingly, for Down syndrome, when you look at chromosome 21, which is a very small chromosome, because the 21 is quite low down. And actually in Down syndrome individuals, they have 3 chromosome 21 instead of 2. Now, normally, if we try to test for chromosome 21, what you do is that you take some cells from an individual you want to test, and then you have a probe. So let's say a probe I label red, and then I have a probe probing the cells from an individual, and then I count how many signals can I get in chromosome 21. So in this case, I can see 3 dots. So meaning that this individual have Down syndrome. Now, we also call Down syndrome trisomy 21. Tri is 3, some is chromosome. So 3 copies of chromosome 21. So now the question is it possible that I can actually do this Down syndrome diagnosis by using those circulating DNA in the mother's blood? So as I've told you just now, those circulating DNA are swimming outside cells. So you cannot actually have this situation of knowing, so how many, you can't see a cell membrane and then count how many of those cell DNA is inside a cell. So how can you do it? So I was thinking, well, maybe it's possible that the babies with Down syndrome will secrete more DNA in the mother's blood. So I tried to measure that, and I did that in 1999. Can see that indeed in people with in pregnancy involving a Down syndrome baby, the level of fetal DNA in mother's blood is a little bit elevated. But on the other hand, you can see there's a lot of overlap with the normal pregnancy. So by using this as a test, it wouldn't be very accurate. So we need something better. Then I was thinking, well, is it possible that a fetal nucleic acid in the mother's blood is a little bit different? Can I find something which is really specific to the baby? And then I was thinking, well, we have many genes in our genome, say 20,000 genes, but maybe the mother and the baby will have different set of genes which are switched on because they function differently. It's almost like when you look at your computer, you might have 10 different programs installed. You have Word, you have PowerPoint, you have Excel, etc. But then only when you start working, for example, with Excel, then you switch it on. So similarly, when we look at the DNA, look at the genes in the babies and mother, not all of the genes are switched on. And the baby will have a different set of genes which are switched on from mother. Now, so basically when a gene is switched on, the DNA will be copied into this thing called messenger RNA, going to carry a message. And this messenger RNA will then tell the cell to make the protein. And of course, nowadays, you're very familiar with messenger RNA because, for example, the BioNTech vaccine is made on messenger RNA. So we're wondering, is it possible that I can actually use the messenger RNA from a baby to test for Down syndrome? And of course, one question is that, whereas we have shown that fetal DNA is present in mother's blood, does fetal RNA exist? So it took us 3 years to do it, and then we showed that it exists in year 2000. So indeed, there's messenger RNA from fetus. The next question is, would some of those messenger RNA from chromosome 21, which is a chromosome involved in Down syndrome? So it took us quite a long time to do it. Imagine I started in 1997. It was 10 years to do this. But in the end, we find that you can see here, the trisomy 21, the Down syndrome baby, have a different messenger RNA profile. You can see this signal and this signal is different from a signal if the baby is normal. So we call a normal baby a euploid baby. But then we have a problem, because when you read the newspaper or yourself have got the BioNTech vaccine, you know one of the weak points of the messenger RNA is very unstable, right? So the BioNTech vaccine will require you to fridge it like -80 degrees. So similarly, if you're testing the blood for messenger RNA, then your blood sample will need to be stored in low temperature, and you need to handle it very quickly. And that, unfortunately, is not very practical, because sometimes the patient will go to the clinic, you take a blood sample and wait for a few hours before you come to a laboratory. So we're thinking, well, messenger RNA is not very good, but then can we go back to DNA? But the DNA has got some problem, because in most of the cases of Down syndrome, the extra copy of chromosome 21 that the baby has got is coming from the mother. As you know, the older the mother is, the higher the chance of that passage of the extra copy of chromosome 21 to the baby. But then, if it is from the mother, then it's difficult, because the baby's DNA is swimming in the mother's blood. So any DNA from mother and baby are difficult to be differentiated if you look at the DNA sequences, the A, C, T, and G, those codes. So we have to move beyond those codes. So those codes is called genetic code, right? So we have to think about what beyond genetic code, there's something called epigenetics. So epigenetics is like your room, the decoration in your room. So even though the walls are fixed, but you can put different paintings and furniture in your walls. So similarly, in the DNA, even with the same ACTG, the genetic code, you can have different chemical compound which can modify the code epigenetically. And for those of you who are doing biology, you know one way is to add a methyl group onto some of the code, the genetic code. But there are many other epigenetic markers. Now, interestingly, for example, you look in my body, my whole body, all the cells have the same genetic code. But yet my brain cells, my heart cells, they function differently. And one of the reasons is because they have different epigenetic modification. And similarly, the mother and the baby have different epigenetic modification. So it took us close to 10 years to also look at this epigenetics to try to tell if the baby have different chromosomes. Now, for example, in this case, we show that if the baby has 3 copies of chromosome 18, I can actually have different signals to a normal baby. So this one we did in 2006. But the problem is that to look at the epigenetic code, you need to use some very nasty chemical. And one of this chemical is called sodium bisulfite. It's very nasty because it actually will kill 90 to 95% of DNA you put in. So there's already not enough DNA in the blood, and yet you want to kill another 90%. So once again, those tests are usually very tedious and not too good at that time when we're testing. So by the time 2007 comes, so I've already been working in this area for 10 years, from 97 to 2007. So we think maybe we need to change some direction and do something else. Now, so you will see actually that in the journey of discovery is frequently like that. So usually you try things and usually most of them don't work. So I would say research is probably 10% inspiration, but 90% perspiration. So you really have to sweat and work hard on it. But on the other hand, even if you know that a particular path doesn't work, it doesn't matter because you already gained some knowledge. And also it's a process which actually counts. Okay, so up to now, we've been spent 10 years trying to see if I can find something which is special about fetal DNA, which I can detect. But then in 2007, I started to think about the whole field differently. I was wondering, is it possible I can actually make a Down syndrome test without having to separately analyze the mother and the baby? So a very fundamental difference in the way of thinking. Now, for example, let's say that you have a wallet, and every day you put in different number of coins into it. Now, let's say I want to know how many coins you have in your wallet. And of course, the most direct way, and if you like, the most intrusive way, is to ask you to give me your wallet, and then I'll open the wallet and count how many coins is in it. And this is a little bit like amniocentesis. I stick a needle into the uterus and then obtain some samples. But a more non-invasive way is to ask you to stand on a balance, right? Now, provided my balance is extremely accurate, if you have 2 coins versus 3 coins in your wallet, maybe I can see the difference. Maybe you're 80 kilogram, 0.01 kilogram, or 80.02 kilogram. So the question is, can I build a balance which is precise enough to do that? So then by the time 2007, we set about building such a balance for DNA. So I was thinking, well, if I can have some ways, I can count the DNA molecules very precisely in the mother's blood. Maybe I can tell if the little bit of fetal DNA has an increase in chromosome 21 sequences. So I was thinking, well, the balance to get is maybe by DNA sequencing. So let's say I have 1 million DNA molecules in the mother's blood, and there's a sequence of 1 million molecules. And because I know the human genome sequence, I can map each molecule to the chromosome from which it comes from. And then at the bottom here, I can do some accounting. So which chromosome have how many molecules? And then see if I can detect that little bit increase in chromosome 21. So we did that, and lo and behold, it worked. So this is sequencing and counting data from a mother's blood. And then you can see in a Down syndrome baby, trisomy 21, if it's a male baby, it's green. If it's a female baby, it's red. And you can see the signal in the red and green are very high as compared with a normal baby, which is a blue and orange signal. Actually, in this group, we did 28 cases. 14 of them have Down syndrome, 14 normal, and it's 100% accuracy. So we did that in 2008, and of course, we're very excited, and we published this paper. But interestingly, within a few weeks of this paper, there was another paper from another group on exactly the same thing. Now, you will find that in science, it's very interesting. Sometimes if you have a really good idea, you can bet a lot of the time somebody else elsewhere in the world also has the same idea. And you'll say why? And the reason is because science is constantly driven by technology. And when you have access to new technology, that other people will have also access to those technology. So a lot of the time you come to this inspiration technology and how hard you work to push it out. And then of course, I'm very curious to see who is this other guy who's competing with us. So when I look at his website, I find there's a guy called Stephen Quick, who is a biomedical engineer from Stanford. And interesting, he also graduated from Oxford, where I studied. But anyway, so with that competition, it means that we have to go quicker. So of course, the next phase is to do a large clinical trial to see if the test is robust and reliable enough on many samples. So we spent the next 3 years doing that. And in 2011, we published that. And so this is the first large-scale clinical trial of this technology. And we find that we can detect Down syndrome with an accuracy of 99.7%, which is actually much higher than I imagined when I first started this journey many years ago. Of course, now with 99.7%, then we thought, well, it's probably good enough to be a test. Because you can imagine that when you do COVID tests, many of the COVID tests are nothing like that accurate. But then being in a university, I couldn't provide a service or a product to help the pregnant woman. I have to work with companies. But when you start working with companies, they will only work with you if you have patents about technology. The reason is because they're going to invest millions of dollars to commercialize your technology, unless there's some ways in which they can protect to stop the technology from being copied by other people. Otherwise, they're not going to invest in you. So then we start learning about patents. And just to tell you what we've been doing with patents. Now, for the last 25 years at CUHK, myself and my students, we have been actually learning how to write patents. And we've got actually reasonably good at it. And the reason is because every year, this journal, Nature Biotechnology, will have a list of the 20 groups in the world with the most patents that year. And interesting, for the last 5 years, my group has been fortunate enough to be on this list. And actually this year, I'm number 7 on the list. And myself and my colleague, Rosa Chiu, so this year we have 18 patents granted. And of course you will say, well, who are the other guys on this list? Now, interesting, the first person is Jiang Feng from MIT, who is actually a pioneer on this technology called CRISPR, which is technology for genome editing. And number 2 is Sahin. He's actually the co-founder of BioNTech. So you can see, the effort that's been putting in. And the third person is Bob Langer, who is actually the co-founder of Moderna, which is another COVID vaccine company. And then interestingly, number 5 is Jennifer Doudna, who has actually got a Nobel Prize a couple of years ago for inventing CRISPR. And then number 13 is another of my colleagues, Alan Chen, also for working in this molecular diagnostics. Now, when you look at this particular list, you can see actually that the Americans have a very strong presence. So different cities like the Boston or like California, and actually there are 3 groups on this list from China and 1 group from Germany. That's a BioNTech group. So you can see that if we want to compete in a biotech space, we actually still have a lot of effort, a lot of work to do. Okay, but that is now. Now, 25 years ago, we're not as good. So I remember when we filed the first patent, our university actually licensed it to a company in UK. And that company actually didn't realize the significance of our work. So they sat on it for a number of years and just throw it back to us and say this is rubbish. So this is another thing that from my experience is that from now, if you have your own invention, I think instead of licensing it to other company, it may be better for you to have your own company. Because after all, if you have your own company, then we can actually develop the technology ecosystem here. So anyway, so we have got the patent back from this UK company. So we have to start thinking about other people who might be interested. So interesting, I then went to a conference in Thailand and met this gentleman who is Dr. Charles Cantor. He's actually the chief scientific officer of a company called Sequenom. So Sequenom was listed on the NASDAQ. And then we start talking, and then he became interested in my technology, and we start collaborating. And then in 2005, Sequenom licensed our technology. But on the other side, in Steve Quick in Stanford, of course, you imagine in Stanford, they have a lot of experience with commercialization. So Steve Quick licensed his patents to this company called Verinata, which was then acquired by Illumina for about 600 million US. And then after that, a number of other companies also launched product on non-invasive prenatal testing, like Ariosa, which was then bought by Roche, and a company called Life Codex. Now, of course, when you have multiple companies playing in the same space, they'll start to fight. They'll start to have lawsuits, almost like a few years ago when you look at Apple versus Samsung or Apple versus Google, etc. And then when they have lawsuit, of course, they will fight the patents. And usually in patents, the first one or the earlier one is the most important. And of course, you imagine in this particular space, the first one belongs to me. So this is quite a famous patent. It's now called the 540 patent. It's a US patent. It's on non-invasive prenatal testing. Basically, it is a method for us to detect DNA of fetal origin that a baby has inherited from a father. And then we can do that just by looking at the blood sample from the mother. So it's non-invasive because from blood sample from mother, and we detect the father's DNA of the baby. And then this patent actually becomes one of the most valuable in the field. So now what this patent allows you to do is this. Let's say this woman is pregnant. You can take her blood and detect things that a baby has inherited from the father. Now, for example, if the baby is a boy, that you're detecting the father's Y chromosome, this fall into my patent. Or if you detect there are two chromosome 21, the one from the father will fall into this patent. And then we license this patent to Sequenom. But of course, the other company want to come in, so they have to destroy my patent. So one of the ways they're thinking about destroying my patent is they use this particular US law called USC, so United States Code 101. So what does 101 mean? 101 is basically a law which sets what sort of invention can obtain patent and what cannot obtain patent. So what cannot obtain patent is things which are the laws of nature. So let's say you discover E equal mc squared. You cannot patent that, okay? Or like natural phenomena. So like you wake up one day, you see the sun, you cannot patent the sun, okay? So people are saying that fetal DNA in maternal plasma is a natural phenomenon, and so you cannot patent that. But interestingly, here I'm not actually patenting the natural phenomenon. The reason why I say that is because if I patent fetal DNA in maternal plasma, it would be any fetal DNA, right? The stuff that baby have from a father or from mother anywhere. But here, no, I'm just saying that the baby stuff that it inherits from a father. And also things that I can amplify using PCR. Of course, a PCR is an artificial process, and that detected by running a gel, right? So it's also artificial, right? It's not a natural phenomenon. But unfortunately, in that particular lawsuit, we actually lost in the lower court in US, and then we appealed to the federal circuit in US. And then Federal Circuit ruled that we lost. And then we tried to appeal to the Supreme Court, but Supreme Court in the US, every year they only have space for about 80 cases. And they decided not going to listen to this case. So in the end, this particular patent, it becomes invalid. And this has a lot of ramification around the world of molecular diagnostics. Because if we cannot get our patent, then many people cannot get a patent as well. So imagine if you discover there's a particular mutation is associated with cancer, and then you try to develop a test for that mutation, then people will say, well, that is exactly the same as here, right? It's a natural phenomenon. Even though you use PCR and stuff like that, you cannot patent. So it's a big thing. And so some people even say that by this, you basically eviscerate the whole patent process. So now, basically, when you file a patent, this 101 issue is a very big thing. But luckily, when we are fighting patent battles, it's like you fight a real battle. We have many layers of walls, many layers of defenses. So this particular 540 patent of mine is only the first layer of defense. So we lose that, but I still have many, many other walls. Now, for example, just now I told you about this sequencing and counting of molecules is another famous patent that we have got the protection for. And then, of course, we've got to constantly keep adding further layers. Now, for example, some people will say, well, you got this counting method, and they're very happy because every time they give you a patent, you have 20 years of protection. But then for me, I'm not happy because I told my student, I said, okay, now you have this counting method. Can you invent something over and above this counting method? Now, so we're thinking now. So counting method is basically, I'll talk about quantity, how much of stuff you have, right? But another way is to say quality. So quantity versus quality. So the quality side is there any qualitative difference between the fetal DNA and the mother's DNA? So I was wondering, how about size? Because the DNA molecules are different, can have different lengths. So is it possible that the mother's and the fetal DNA might have different lengths? So I told one of the students to measure the length. So the x-axis here is the length of a molecule, and the y-axis is how much those molecules we have. And then the blue lines is the fetal DNA, and the red line is the mother's DNA. Now, interesting, you can see the fetal DNA is on the left-hand side of the mother's DNA. So meaning fetal DNA is a bit shorter. So then it's interesting. That means that we have some short fetal DNA and have some long mother's DNA, and you normally mix in the mother's blood. Now, so if that's the case, then the more fetal DNA you have in a sample, the shorter it would be. Now, for example, like here, you have this sample, this dotted line is a sample of 20% fetal DNA. And the bottom one is a sample with 10% fetal DNA. So you can see when you look at the short molecules, like over here, you have the one with 20% will have a higher peaks. But on the other hand, if you look at the longer side, then the pattern is reversed. So because of this, then our student then developed this thing called the size ratio, which is basically tell you how much is the size of the fetal DNA. So it's just like that. So the size ratio is short over long, very simple. And then you say, well, what is the use of the size ratio? Now, interestingly, basically the size ratio will tell you in a particular sample how much fetal DNA you have. So the size ratio is on the y-axis, and the x-axis is how much fetal DNA. You can say I can predict it. And this is already a breakthrough because the number one determinant of this accuracy of non-invasive prenatal testing is how much fetal DNA you have in a sample. It's almost like nowadays when you talk about your COVID test, you want to know the CT value, right? The lower CT, the more COVID virus you have got. So similar here, if I'm a pregnant woman, I come back this test, I like to know if my fetal DNA concentration is high, then I know my test accuracy is higher. But before our invention, you can almost easily do it if the baby is a boy, because you can measure the Y chromosome, just like here on the bottom. But if the baby is a girl, that's more difficult. But now, by using this size ratio, you can do it irrespective of the sex of the baby. And then you wonder, what else can I do with this size? Then I'm thinking this way. So imagine that you have this mother's DNA is long, and then you have a fetal DNA which is short. And normally they mix together in mouse blood and give you a size ratio. So now imagine if the baby have Down syndrome, it will have one more copy of chromosome 21, which is releasing a short DNA in mouse blood. Then theoretically, in the mouse blood, the chromosome 21 will have a shorter size distribution than other chromosomes. So that's the theory. Is that the case? Indeed, it is. You can see, for example, like you look at chromosome 21, look at the size. If the baby have Down syndrome, the T21, so trisomy 21, then the distribution is very different from the other cases in the normal. But if you look at chromosome 18, then if the baby have 3 copies of chromosome 18, then the size ratio is different. But on the other hand, if you look at chromosome 13, then once again, if the baby have 3 copies of chromosome 13, then the size ratio is different. So in other words, by using a size, you can basically do different things from the counting. So 2 things are separate. So interesting, my student who actually invented this pattern, when she graduated, she actually can use the patent income to buy an apartment in mainland China. So to show you how useful it is if you can actually invent new things. And you should not be happy just because you have invented previous counting. You always will push forward. Now, just now I told you when you have patent, who is the first is the most important. But the question is, how do you classify who is the first? Now, interestingly, for most part of the world, it is who filed the patent first. So let's say that I go to the patent office today at 10 a.m. I filed a patent, and you come at 10:30, then I beat you. Now you say, well, that's very reasonable, right? But interestingly, not everywhere in the world do this all the time. Now, for example, in the United States, before 2012, they have a different system. They use the first to invent. And then you say, well, what's the difference? The first to file, first to invent. The first to invent is different. It's like last night when I'm having dinner, I suddenly have an idea. And let's say you have breakfast this morning and you have the same idea, then I win because I had the idea first. But the question is, how can I prove to the patent office that I thought about idea last night in dinner? This usually you don't write down your idea. But interestingly, some people do, right? So there has been some crazy thing about people inventing something, they write on their fridge and then take a picture of the fridge, and that is used as an evidence, or your email or whatever. But you should imagine to decide who invent first will take a lot of effort. And so because of this, in America, they have a special type of lawsuit called interference to decide who wins first. Now, so you can imagine with me and the Stanford professor, both inventing accounting methods, then of course we'll fight some interference law battle. Now, when you fight lawsuit in the US, it's very funny. For example, they have one process called deposition, which sometimes when you read about in US newspaper, basically what is that process? It's a process whereby the lawyers on the other side can interrogate me. And every time they can interrogate me for 7 hours. So basically you go in a room and they have a clock and you sit down and the lawyers are on the other side. They'll take a video of you, they'll record what you say. And they even have people dial in on teleconference listening from what you say from the other part of the world. And then let's say that you want to go to the bathroom, then they'll stop the clock and you go to the bathroom, you come back and they start the clock again. They'll make sure they have their 7 hours and they can interrogate every time for 2 days. So you can imagine that is a very tough process. I've been through that process many times. Actually, my lawyers told me that if you're in a deposition, it's like in the football, in the penalty shootout, and if you're the goalkeeper, you can only lose the game. You cannot win because you are not the guy who is going to kick onto the outside. You're only defending. So basically, you have to make sure your 7 hours a day, you do not lose the goal. So that's very, very stressful. But anyway, so after a lot of stress, the two sides, Sequenom and Illumina, decide to settle. And then they pay us some money and then we pool all of the patents together. And then the technology was launched in October 2011. So now the technology is called NIPT, Non-Invasive Prenatal Testing. It's now actually used by millions of cases every year. And actually the royalty from this project probably account for maybe over 90% of the royalty income of our university. And then you say, well, what about Hong Kong? Now, because I'm born in Hong Kong, grew up in Hong Kong, I want to make sure that NIPT is a technology invented in Hong Kong, that we have a very good service. Now, our technology is funded by the Innovation and Technology Fund. So this is Ms. Janet Wong, who is the previous commissioner of the Innovation Technology Fund. So one day I met her and Janet told me, say, Dennis, why don't you think about having your own company to provide NIPT? And then I thought about it and then went and talked with some of my colleagues. And in the end, 3 of us, myself and 2 of my colleagues who are my previous student, so Rosa Chiu and Alan Chan, we decided to actually open a company. And actually the company, we call it Excelom, which stands for extracellular genome. Now, the Chinese name of it means Nga Si Lang. And Nga Si, we thought because we are Manyan Nga Si, we are scholar. So even scholar can do it. And then Excelom actually becomes a market leader of NIPT in Hong Kong. And then we also actually give part of our patent free of charge license to a hospital authority so that they can actually provide a free service to some of the highest risk pregnant women in Hong Kong. And of course, you imagine then the thing is going smoothly until suddenly we find that somebody is copying our technology in Hong Kong. And then Excelom then decided to actually sue that other company, and we filed a lawsuit. And then we realized actually that there's some problem. Now, whereas Hong Kong has a very well-developed legal system, but the problem is that very few people actually fight patent lawsuits in Hong Kong. Now, there's some people who fight copyright lawsuit, like the music copyright, games, and sort of stuff. But actually, patent is a different thing because previously to now, we have relatively few technology startups in Hong Kong. So as a result, the judges and the lawyers are not very familiar with this. Now, for example, I remember when we fought the lawsuit in Hong Kong, there are only 3 senior counsel in Hong Kong, so senior barrister, who actually have experience with patenting. So imagine my company got 1 and the other opponent have another, and the 3rd one is about to retire. So you can imagine that was the situation. And also I remember our case actually went to a particular judge, but the judge actually sat on our case for a few weeks and decided not to try it. And our case went to another judge, and that judge once again decided not to try it and went to a 3rd and went to a 4th before we have somebody to preside over the case. And also, we also find that fighting lawsuit in Hong Kong is very expensive. So we fight for 4 days, and it costs us 15 million Hong Kong dollars. So imagine for a startup company, it's really very, very bad. So basically, I think that if Hong Kong want to be a technology hub, we probably have to improve in this area a lot. So after Excelom become a market leader, then we say, okay, can we serve the citizens in mainland China? And then actually we find that there's some difficulty. And the difficulty is because actually currently there are restrictions in the transfer of DNA samples and data across the border. So we find that as a company in Hong Kong, I cannot actually serve clients in mainland China. And then we thought, well, how about if I go into, say, Shenzhen or Guangzhou to open a branch? And then I find that I cannot. And the reason is because for a certain high technology area, like genetic testing, if you are not a mainland citizen, you cannot actually open a company yourself. So I couldn't do it. And the 3rd thing I thought, well, how about if I have patents in Hong Kong, maybe I can license a company in China. But answer is you cannot. The reason is because patents granted in Hong Kong have no enforcement in mainland China. So basically, I think that those 3 issues will need to be solved if we were actually going to be one of the engines in driving technology in the Greater Bay Area. Of course, in the future, there's some possibility. For example, like the Hong Kong government is now building something in this thing called the Lok Ma Chau Loop, which is an area between Shenzhen and Hong Kong. Now, if we can make this loop area to be a very special zone where DNA samples and data can go in and Hong Kong companies can move in there to provide service, then maybe this will be a beginning. And then up to this point, you can see that prenatal testing seems to be going pretty well. And then you say, well, what else can we do? And then I was thinking, okay, now you can remember I discovered fetal DNA in maternal blood by looking at male DNA in a woman's plasma. And then I was thinking, is there any other situation where a woman will have some male cells within her body? So after I thought about it for a while, then I wonder about transplantation. So let's say if I donate my kidney to a woman, is it possible that my kidney will release my male chromosome into the recipient's blood? Then I did that. And lo and behold, I find that. So we discovered this in 1998, the presence of donor DNA in a recipient plasma. And then, of course, you say, well, what is the use of this? And the use of it is that the DNA is normally inside cell. The reason that a cell will eject the DNA out is because the cell dies. And of course, in a transplantation context, the cell will die if the graft organ is being rejected. And so basically now, this becomes a field in which by measuring the donor's DNA in the recipient's blood, you can actually monitor rejection, you treat, and it comes down. And there's a number of companies in this space. For example, this company called CareDx in the USA. And of course, you can see this is generalizations. In other words, you've got to be able to see the connection between apparently unconnected field, like pregnancy and transplantation. And similarly, when we're now looking at blood plasma, this fluid, then you've got to think, is it other fluid in the body which you can also use it? And of course, one other fluid would be like urine. Now, for example, you can imagine the urine is produced by the kidney. If the blood of the pregnant mother have got a fetal DNA, is it possible that some of the fetal DNA will be filtered by the kidney and enter in the mother's urine? So we tested that and find that to be the case. So in other words, you can actually do even more non-invasive testing by looking at the urine of the pregnant mother. And there's a lot of other interesting experiments you can do now. For example, I talk about transplant. So you can imagine what happens if you have a woman in which the transplanted kidney is from a man. So the plasma of the woman is female, but the kidney is a male. So is it possible that the kidney will somehow also add some male DNA into your urine? And then another situation would be, let's say the mother, a woman, has a bone marrow transplant. So in this case, the plasma is male, but then the kidney is female. So in this situation, what would happen to urine? Is a urine male or female? So imagine by making those combinations, you can do some very interesting and enlightening experiment. So basically what I'm trying to show you is that you need a lot of lateral thinking. From thinking of one thing, you need to go and translate into another dimension. So parallel thinking. And of course, if you think about urine, then once again, you can think about how about other body fluids, saliva, tears, sweat. You know, so basically, is a lot of other possibilities. So in summary, I hope I've convinced you that now, if you want to do innovation, if it's something worth doing, like non-invasive prenatal testing, which people have been pursuing for over 50 years, then by definition, it is difficult. Because if it's not difficult, other people will have done it for you, okay? So remember, it's actually the process is as enjoyable as the result. And then also it's important to actually look at problems from multiple angles. You can see that when I try to solve this problem, I was looking at the total amount of DNA, I was looking at RNA, I was looking at epigenetics, and now looking at counting, so different things. And then you can also see there's a synergy between academic research and commercialization. Because if I only do research, then my discovery only present in the library, in journals, online, right? But then if I don't work with a company, you'll never have a product in the clinic. And also if you want to work on commercialization, then be familiar with the patenting process. Because I found my first patent when I was a medical student, and I'm still learning it. So I'd like you as a student to start learning your patenting skills. And finally, if Hong Kong would like to become a technology hub, there's a lot of streamlining of policies, like the samples, patents, etc. So finally, I'm very lucky that over the last 25 years, I have a really good team with me, and I'm most grateful for the comradeship and cooperation. So thank you very much for your time.
So thank you, Dennis, for this very inspiring talk. Now our MC is going to start the Q&A sessions. Yes.
Thank you, Professor Chen. Let us express our gratitude to Professor Dennis Lowe for his wonderful lecture with a round of applause. May we invite Professor Chen to hold the Q&A session. Students and staff who have questions, please click on the Q&A button on webinar and submit your questions. Both English and Chinese questions are welcome. The moderator and the speaker will read and answer your question one by one.
Dennis, excellent talk. You know, again, learn a lot of your experience and all your hard work, both in academic research and also in business. You know, to me, that is a very strange territory. Now, I think the students learn a lot as well. I mean, they are now living in a totally different world from 20 years before. And I think you have inspired them to think about the future, about the career from different angles. I started to receive quite a number of questions. Maybe I start asking the first questions here. The first one is, would you consider yourself as an entrepreneur? In other words, how do you balance the role of researcher and entrepreneur? So what would be your response?
Actually, I think that with life is a process of learning and change. Now, for example, like when I was in secondary school, I was very interested in computing. And then when I went to medical school, I'm studying. And then, you know, so basically a different phase in your career, I think you do different things. And so actually currently, I think that entrepreneurship is important because I believe actually this is for example, a future for Hong Kong. So we need to develop a knowledge economy. And for the young people in the audience, we need to actually create new career opportunity for all of you. And actually having a technology ecosystem is one way to do it. And so I feel that it's my responsibility to actually use what I've learned before and try to create some of those ecosystems. Yeah.
All right, yeah. The second question, you know, it says, thank you, Professor Lowe. The NIPT is now very commonly used in mainland China. So are you collecting any royalties from the mainland?
Okay, interesting. So it's because of those three issues, actually currently, I've not actually made any income from mainland China. So that's why I think that if we're going to have this Greater Bay Area and have Hong Kong to have a role to play there, we need to actually streamline those areas. So basically, I'm collecting royalty from all over the world, except actually our closest region. So I think we need to do something about streamlining this. Because, for example, I could have competed in mainland China if I'm allowed to open my own company in mainland China, but I couldn't do it, and we could not collect the samples, and then we could not enforce the patent that way. So I think I really hope that the government, we should actually work with the authority in the mainland to try to actually streamline this process. Yes.
Yeah. Okay. But to me, that is well, have you ever tried to partner with a institutions or a good friend in mainland China, and then file or submit a patent applications in mainland? No,
actually, of course, I've filed patent applications in China. Of course, I have. Yeah. So, but I think that direct way is that I need to have a presence. Basically, we need to open my company there. So as a matter of fact, we are doing that now. But the problem is that if you want to open a technology company in mainland China in one of those sensitive technology areas, like genetic testing, you use something called VRE, variable interest entity. Okay, so the problem is that VRE structure is a gray area. So my thinking is that if we are encouraged, if the Hong Kong scientists and entrepreneurs are encouraged to have a role to drive the technology, then we should not have to resort to using gray structure, right? So that's why I think that the basic policy really need to be streamlined.
Okay, yeah, great. And next question is how to patent lawsuit in Hong Kong. What is that in Hong Kong ended? How the patent lawsuit in Hong Kong ended, and did you just let it slide?
Okay, that's a very interesting thing. Now, so basically, what happens is that in the first lawsuit, we sued the other party, want to have a preliminary injunction, so we didn't get it. And then in Hong Kong, they will then ask you to go for mediation, 調停, right? But then we find that 調停 to be not very useful. But interestingly, maybe lucky to us, the other party was going for IPO, trying to be 上市. And so if you want to go for IPO, you have to settle all lawsuits. So basically, they just have to settle. So you see that if you run entrepreneurship, actually sometimes the environment is very important. So it changes every day. Anything can happen, right? So you need to be very alert and then just move with the flow. Yeah.
And sometimes it relies on luck.
Exactly.
Yeah, when the right moment comes, then you win. But can I ask next questions? Are you working on non-invasive diagnosis for other diseases now?
Yes. So actually, I would say that nowadays, my work probably 50% is on cancer, 50% on fetal. And we also use some very interesting new technology. So recently, for example, we have also have artificial intelligence techniques in our work. And we find actually those to be very, very powerful as well.
Right. There's another question from students. Do you think Hong Kong is an unfavorable environment for scientific research and development?
Well, I would say that every place, there are pros and cons. Now, for example, I used to work in Oxford, but I came back to Hong Kong 25 years ago. I find that actually over the last 25 years, the level of funding in Hong Kong actually have increased a lot. Now, for example, when I first came back, a grant is probably like 500,000 Hong Kong dollars, right? That was 1997. And then gradually I see that with the development of the Innovation Technology Fund, then I start to get grants which are about 4 or 5 million. And then after that, you can see there's some bigger grants like Area of Excellence, which is about 15 million, that sort of thing. And recently with this InnoHK thing, I'm actually obtaining over 400 million. So in terms of grant, I would say that we are actually rising very quickly. But then, of course, if you're talking about the workforce, I would say that still some of our best students will tend to like to choose things like medicine or law, and still relatively few will choose science. So which I hope in the future will change.
All right, yeah. Another question, probably from outside, not the university. She wants to ask why are you interested in biology? Very simple question.
Actually, I mean, when I was a student in secondary school, I'm actually interested in biology as well as even engineering, computing. I was actually accepted by Stanford to do electrical engineering in Stanford. But in the end, I decided to go to Cambridge to do medicine. So I'm interested in science in general, but then I thought, well, biology is complex, because the 20th century is the century of physics, but I believe the 21st century is the century of biology. And so, yeah, so I think the last 30 years I've found it to be very exciting, and I haven't regretted doing that. Yeah.
Yeah. Next questions is concerned about the prospect of scientific discoveries and the process regarding patents in Hong Kong. Are they, for the development of this, are they cheerful in the future? Or maybe around 10 to 20 years later?
I would say yes. I mean, for example, like previously, we are not even in the top 20, but at least now for the last 5 years, we now start to see Chinese groups in there. And hopefully, if you look at the number of publications from China and number of patents filed in China, is actually increasing. So maybe in 10 years from now, that we will even feature more prominently. So yes, I'm positive, and that's what we are training our students to do. Some of my even first-year PhD student or even medical student, if they are in my lab, they will actually learn to write patent even right in the beginning.
All right. Another questions. First thing to do is thank you for this wonderfully engaging talk. Do you think being a successful researcher require a business mindset? And is it productive? Is it productive to perceive research endeavors as similar to running a successful business in a competitive market? Furthermore, do you think this creative spirit is something which can be learned?
Yes, I think that there's a lot of similarity between running a research project, research group, and running a company. So for example, you both will need to have the funding, you need to get some money, and then you need to, control how that resources is spent. And then you have to have a timetable to do it. And of course, I mean, in the old days, us academic are probably more relaxed on the timetable than in the companies. But now that I've worked with companies, then usually if you're in companies, they have a project, they will, of course, very tight timeline, right? You have got to finish it in 2 weeks, etc. So in a way, I'm running the lab a little bit like that as well, which I think is also useful. And whether how you learn it, I would say that you probably through practice. I mean, first you start with small ground and gradually have more. And also I think it's useful to have a mentor who has actual experience in doing that.
Right, yeah. Another question, probably from a postgraduate student. Can a fresh graduate have the similar approach, like starting a company based on my own final year project outcome? Or the most ideal approach is to get a PhD degree before I start the company?
I mean, to be honest, I would say that the PhD, I mean, all those degrees are names. The most important thing is what experience you have got, right? So for example, if you are doing a final year project, and even though you don't have a PhD, but if you actually have a good idea, and you talk with all the experienced people about, you've got to make sure, look at that idea from multiple angles, and it's still a good idea. Then I would say that PhD is not essential, right? I mean, look at many of the successful people like Bill Gates, you don't even have to finish your bachelor degree to be successful. Yeah, but on the other hand, you also have to have some negative story to remember. For example, for those of you who have heard of Theranos, right? So you also got to think that in that scenario, maybe the founder has not been sufficiently trained in science to perceive all the, issue about scientific research. So I think you just have to first know your limitation and know your strength, and be humble all the time. Yeah.
Right. Yeah. And other questions? As a researcher and entrepreneur together, how do you balance the research benefit, the research that benefits the most patients, especially the poor patients who cannot be benefited from the technology, and also profit making for your company? How could you balance these two? And perhaps using the incomes to further support your ongoing research.
Yes, so I think this is a balance that everybody has to make. What is your balance? So in my case, for example, as I told you, from the hospital authority, you give a proportion of the licensing rights to a hospital authority free of charge. So that bit, we are actually not making money from it. And then also under current Chinese university rules, if you make an invention, if have income coming back, some of it will go to you personally, but some of those will go to your department, and some of those will go to your university. And so in a way, those will also help you to educate further student, etc. So I think it's a good balance. And also that income to the department, I find is very useful. The reason is because, let's say if I don't have that income, if I have a good idea today, I have to apply to the government for a grant, and that could take 1 year, right? So I mean, 1 year later, I may have a chance to do a research. But now I have royalty income. Anytime I have an idea, I can do it tomorrow. And that is important. And time is of the essence.
Right. Another questions from the guest. She would like to know how you keep generating new ideas and keep creativity alive. How much time and where do you spend learning new knowledge every day? I do want to know as well.
Actually, I think different people have different creative cycle. Now, for me, I need to be very relaxed before I can be creative. So usually I'll be doing something unrelated. I mean, for those of you who have heard my first talk, one of my good ideas I heard when I thought about it when I was watching a Harry Potter movie. And also, I mean, believe it or not, I like to play video games. So sometimes after I play video games, I have ideas as well, because I'm very relaxed. So yeah, so I would say that don't crowd your brain with too many things. Don't be too stressful. Just be relaxed. Do what you enjoy the most, and then suddenly you'll think. And also another thing is that I like to talk with people. So talking with people, suddenly you also will push your mind to start thinking.
All right. So another questions from a guest. How do you relate your studies to what my former schoolmates, Dr. Chui Laoji? I guess, you know, he want to know whether your experience has some similarity with Professor Chui.
Interestingly, I like to study the stories of famous scientists. Now, to me, each of those stories is enlightenment. So I remember when I was a medical student in Oxford, I actually read about Professor Chui Laoji's work. Now, because that's interesting, because now, of course, we know Professor Chui has won the competition. But actually, in that time, there's a number of people who is competing with him. In London, there's a professor called Professor Robert Williamson. And then they're competing neck to neck. So each time they find a new marker, then they're both sides. And I remember, actually, there was one time when Professor Williamson actually talked about a gene he thought was cystic fibrosis, which turned out to be the wrong gene. So that was a very, cliff-hanging moment. And of course, and I still remember when Professor Chui Laoji published the cystic fibrosis work in Science in 1989. There are 3 papers back to back. That was a very unusual, probably the first time ever in Science. And so I'm very grateful to have a chance to meet him. So, yeah.
So next question from students. For medical students or medical professionals, would you give some suggestions to us regarding how to connect our professions, medicine, to business areas? Any preparation or work we need to do?
Now, so I would think that, now, for example, like my talk today, at least you will hear this is the the material that you need to have a business. So you need, for example, to have patents, and how do you protect your patents? So you need to plan now, because, for example, like some of my colleagues with not so much experience, sometimes they will go on a conference, okay? They want to talk about work, and suddenly when they remember, they haven't filed a patent, and then they're in trouble. Because remember, if you talk about your work in public without filing a patent, then your patent is gone for most part of the world. So you don't want to make those mistakes, right? And then you also see, for example, like I told you about filing patents, you file the patents, and then 1 year later you can file again. But as you can hear from my competition with a Stanford professor, competitions can be very tight. For example, some of my work with a Stanford professor, it only separate by weeks. And so in that situation, that means that 1 year gap is not good enough. So you have to keep filing. So if I was the medical student who asked the question, I would start to read patents. So instead of just reading the paper, also read the patent associated with it. Because it's a different game. Because a paper is only say that what you have done, but a patent is what you predict is going to happen in 20 years. So it's a different game. So which is fascinating. And also another thing which I would suggest is that writing a patent, the writing skill is very important. So just because we are scientists doesn't mean that we do not focus on our literary skills. So also try to enhance that as well. And then I will also think about, for example, subscribe to some internet newsletter. Now, for example, if you're interested in biotechnology, there's something called STAT, S-T-A-T, STAT News is very good because they talk about all the new drugs, new tests. And then in my case, in genomics, then Genome Web is very good. So read about those things. And Nature Biotechnology is also very good because sometimes they talk about some important patterns for you to study. Yeah.
Okay. So maybe ask another questions. There's a question about the depositions process. Now, could the deposition process in USA hinder researchers from making progress which favor the opposing researcher? And could the wordings in the patents affect who has the final patent on the technology? Like if the second patent mentions something which the first patent doesn't, will that affect the decision from the judge?
Okay, now deposition is a very interesting thing. So when deposition was created, it was meant to be a cheaper way for people to fight lawsuits, because you can imagine that's just me facing the lawyer on the other side, rather than me having to go to court to face the judge, okay? But unfortunately, as the current way is run in the US, I find deposition become very tedious. And so, for example, before they do a deposition, someone will come to my office and get off my computer that I've been using for the last 15 years. Every single laptop which I've used, they'll make a copy of the hard disk. And when they go back home, they'll have a whole team of people go through every single message I've got, every single email, every single Word document. I remember the day when I went to a deposition, the lawyer at the outside said, well, I have 6 million pages on you. And then suddenly take out this page and say that on this day, you send this email to X. What does this mean? So basically, you have to be very careful. If you're going to fight a lawsuit in the US, what I would suggest is that all your email, all those writing, you assume that eventually will go to become 呈堂證供.
Yeah. Because we are short of time, that we could entertain, I think, 2 more questions. Let me ask a simple one. 你有冇試過放棄 biology? Have you ever thought of giving up biology?
冇嘅，因為我覺得其實我係 interested in 嗰個 process， 即係我個興趣係嗰樣嘢，即係我其實當我嘅研究咧係一個嗜好嚟嘅，即係等於好似我譬如打 tennis， 我唔係好叻，但我係 enjoy 個 process。 咁即係等於研究。就算我譬如話，即係你見到我10年嗰啲嘢咧，好似係好多都唔 work 嘅。咁但係其實 that is the nature of research。其實唔係嗰個結果咯，而係嗰個 process。即係每日我會翻嚟同啲同事一齊傾，或者學生一齊傾，傾完之後唔 work，咁又諗下下一條橋咯。咁其實係好 interesting 嘅。
At a certain stage of your career, you might come to a point that you need to consider whether you should go totally to business or totally to academic.
Well,
at this point, what are the criteria that you will be thinking of?
Well, usually I will do the thing that I'm best at, okay? Because I believe each of us probably have some intrinsic strength. Now, for me, even in business, I actually do the new technology side. So I invent new technology. So even if I'm full-time working in a business, I'll probably be a chief scientific officer. I won't be a chief executive officer. I won't be a CEO, okay? But I will be a CSO. And of course, on the other hand, some people are very good to be CEO. So I think each of us has to be humble and aware of our limitation as well as our strength, and do the thing that we're best at.
Okay, thank you so much. Yeah, I think we need to stop right here. Thank you again, Dennis, for giving us such a beautiful talk and very inspiring, and also answer so many questions in the Q&A. I think we have received the largest number of questions in the assembly that I have handled so far. So thank you so much again, Dennis. We are expecting you to share with us your experience again in the third lecture.
Thank you.
Thank you.
Thank you. Bye-bye.
Oh, bye-bye.